Hello and welcome everybody to my channel ominibioentrance, and today we will be seeing CSIR PYQs of June 2023 Shift 1. I will be dealing with applied biology and methodology, DNA replication, protein synthesis and genetics. Then next one. The following statements are made about telomeres. Which of the following option represent combination of all correct statements? Telomere binding proteins are believed to shield telomeres from the cell's DNA repair machinery, preventing them from being recognized as double-stranded breaks. That is correct. Telomeres in human cells are repeats of TTAGGGG sequence that can extend up to 150 kb, which are replicated by the action of TERT in actively dividing cells. Yes, telomeres in human cells are repeats of TTAGGGG sequence that can extend up to not 150 kb, it is 15 kb. So this statement is not correct. B is not correct. In differentiated cells, telomerase is inactive, leading to shortening of telomeres over hundreds of cell divisions, damage to ends of chromosome and eventually apoptosis. That is also correct. Then next one. The persistence of telomerase activity in several cancer cells allows the cells to continue to proliferate. That is also correct. Which of the following option represent combination of all correct statements? So A, C, D. That is option 4. So the answer here is option 4, A, C and D. Then next one. T4 phages were plated on 3 E. coli bacterial plates labeled 1, 2 and 3. The phenotypes obtained are depicted in the picture below. The black spots represent plaques. These are large plaques, these are small plaques, here no plaques. The following combination of conditions were given to explain the results. This is plate, bacterial strain, R2 locus. From the options listed below, select the one that accurately lists the E. coli strain type and the corresponding R2 locus type. So first one is this, plate 1 is this, you have got large plaques. So large plaques, where do you see in this table that I have created? It is large plaques are seen for E. coli B in R2 mutant. So first one, then it will be 2, that is E. coli B, bacterial strain will be E. coli B and it is an R2 mutant, so B. So first one will be 1, 2, B. Then next one you have got small plaques. Small plaques are seen in wild type T4 phage strain infecting E. coli B and E. coli K12. So first one, second one, 1 and A. So second 1A. Then next one, third one, no plaques. No plaques are seen when R2 mutant T4 phage strain E. coli K12, no plaques. So this is E. coli K12, 3 and B mutant. So the answer here is option 2. Then next one. A researcher isolated chromatin from mammalian cells and digested with micrococcal nuclei in different tubes for 1 hour, 3 hours, 6 hours and 12 hours. So what is micrococcal nuclease? It is an exoendonuclease and can digest DNA and RNA. Thereafter, DNA was purified from all the digested chromatin samples and 2 independent southern blot hybridization experiments were performed with probe 1 and probe 2. So after digestion, the DNA was purified. All the digested chromatin samples were subjected to 2 different southern blottings. Probe 1 was used for one blot, probe 2 was used for the other blot. The probe 1 and probe 2 correspond to different loci of the chromosomes. The images below represent the southern blot hybridization pattern generated by probe 1 and probe 2. Now, if you see here probe 1, there is not much difference in the band intensity because the level of digestion is not high in this region of the chromatin. Whereas here, if you see, the band intensity is decreasing, showing that digestion has taken place or digestion is increases with time. Following statements were made to explain the results of the southern blot experiment. Which of the following options represent the combination of all correct statements? Size of probe 2 is smaller than probe 1. That is not a correct statement. Then probe 1 may correspond to the centromeric region of the chromosome. Now I told you that probe 1 there is no much difference in the band intensity because the level of digestion is not high in this region of the chromatin. Here if you see there in probe 1 case, there is not much difference in the band intensity when compared to probe 2. This is because it is highly condensed region corresponding to heterochromatin region. Okay. So probe 1 corresponds to centromeric region of the chromosome. Centromeric region of the chromosome is heterochromatic. So probe 1 corresponds to that region. It is true. So B is correct. Probe 1 may correspond to the hypomethylated locus of the genome. No, it should be hypermethylated, not hypomethylated. Then next one. Probe 2 may correspond to a euchromatic locus of the genome. Here probe 2 if you see, I already told that degree of digestion increases as time increases. 12th hour the intensity of band is very less. This is because it is less condensed region of the chromatin corresponding to the euchromatin region. So D is also correct. So B and D are correct, whereas A and C are not correct. So the answer here is option 2, B and D only. Then next one. Given below are terms related to various experimental techniques in column X and their application column Y. Which one of the following options represent all correct matches between column X and column Y? Mass spectrometry. Mass spectrometry is for identification of post-translational modification of proteins. Pulse field gel electrophoresis is separation of whole chromosomes. Isoelectric focusing is separation of isoenzymes, and PAC-bio is single molecule real-time sequencing. Answer is option 1. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Previous year CSIR questions also I have uploaded. You may go through that in my channel. Please do like, share and subscribe if you are new to my channel.